My name is Tim Freeman, and I am a PhD student at the University of Sheffield. I am excited to be presenting today my poster titled Identifying Genomic Regions Susceptible to Systematic Sequencing Error to Improve Variant Detection, which is about a quality control tool I have recently published in Genome Research to flag up systematic sequencing biases. So first of all, why did I want to identify systematic sequencing biases? Many fields of biology and medicine rely on accurate sequencing. Identifying biases enables my tool to filter out false positive variant calls from sequencing results, which may be missed by standard quality control processes. In addition to this, identifying systematic biases in sequencing can help in characterizing the source of these errors, and this information could potentially be used to optimize sequencing parameters, improving the overall accuracy of genomic sequencing. Displayed on the figure here is an example of how this work can provide real benefits to doctors and researchers analyzing sequence data. This figure is an image of IGB viewer output showing the genetic sequence within the SOD1 gene in an individual. At the colored locus, about 16% of reads suggest the presence of a G allele, shown in orange. If there was indeed a G allele present at this position, it would be diagnostic of this patient having ALS, so it is very important for a clinician looking at this to know whether this G variant is a real variant or an error. Using my method, I have discovered that reads providing evidence for the G allele occur at a similar proportion to this across every single human sample sequenced using this same pipeline. So in this instance, I can identify that this is in fact a systematic error and we can avoid making an incorrect diagnosis. In a similar way, I can catalog all of the alleles that show the systematic bias as suspect loci across the entire genome and provide this data to scientists and clinicians who might have any of these variants in their own sequencing data so that they can avoid false positive results in their own analyses. To provide further background to this problem, we also need to ask whether existing quality control processes can correctly identify and remove these false positive sequencing results from our data, because otherwise our method wouldn't provide much additional value. The methods listed on the left can help with QC, but we have found that all of these are very limited in their ability to filter out systematic false positive variant calls and have other drawbacks, which our method addresses. So to summarize all of this, here are the aims of the study. We wanted to identify and catalog all of the systematic sequencing biases we could find across the entire human genome, analyze how frequently these occurred across genomic positions, and check if they were interfering with the accuracy of variant calling, especially within clinically important regions such as clinical panels. The final aim of the study was to develop and share the results and method for this so that scientists and clinicians could use these findings to improve the accuracy of their own genomic sequencing data. So how exactly does this method work to identify suspect loci across the genome? First of all, I needed to take genomes from large cohorts of patients, in my case ranging from 150 to 215 patients, sequenced using the same pipeline. For each of these patients, I looked at which allele every read provided evidence for every position across the genome, and I computed the relative allelic fractions. I used these as the input to obtain the aggregate allelic fraction values and the standard deviations across all of the patients. Together, these were saved in a data structure we have termed an incremental database, or INCDB for short, which removed any confidential patient information. I compared the standard deviation values for each allele with their given aggregate allelic fractions on a density plot. I generated a Monte Carlo simulation to identify the lower bound for expected standard deviation for each individual allele across the genome and discovered a large plume of alleles occurring at low to medium allelic fractions which had very low standard deviations, indicating that these were systematically occurring at this level across the cohort and were likely to be false positives, which we termed suspect loci. After we had identified these, we finally examined where they were occurring along the genome, what regions they were enriched in, and whether they were resulting in any actual false positive variant calls in clinically important regions and panels. So did these biases actually affect real variant calls that were being made? For figures A to C, we varied the reference genome and alignment algorithm that were used to identify how this would affect the level of systematic bias observed, and we found 1 to 3% of all genomic positions annotated as suspect loci depending upon this choice. Even though 1 to 3% might seem like a low level to observe systematic bias at, it accounted for millions of positions across the human genome, demonstrating that our method caught many false positive alleles that otherwise would have gone unnoticed. In figure D, we looked at the actual single nucleotide variant calls that occurred for suspect alleles in an independent genome. We observed on the order of 200,000 suspect SNVs for each pipeline, which were likely to be false positive variant calls, including many within introns, exons, and clinical panels. We also checked that these suspect SNVs would systematically occur at a low level in a patient independent from our cohort, who had been sequenced using the same pipeline. On this figure, we show that SNVs that were not at suspect loci had allelic fraction peaks at around 50% and 100%, corresponding to real heterozygous and homozygous peaks. However, suspect SNVs had a consistent low-level allelic fraction peaking around 20% rather than showing correspondence to real biological peaks, thus confirming our hypothesis. I have established that systematic biases are widespread throughout the genome, including within clinically important regions, and catalogued all of their positions for other scientists to use. If we want to identify the source of these biases, however, it helps to identify where these positions are most heavily enriched, since this gives us clues about whether these biases arise more on the sequencing machine or read alignment side of sequencing. Unsurprisingly, we found that systematic biases were most heavily enriched within problematic regions, especially within the flanks of large homopolymers. This is likely to be because the repetitive nature of the bordering regions can cause a low proportion of reads to map to the wrong position in every patient. Judging by this, and the other regions that are most heavily enriched in suspect loci, alignment errors seem to be a bigger source of systematic biases in general, rather than the sequencing chemistry side of things, at least when using Illumina HiTSeq sequencing data. Comparatively, suspect loci occur at much lower rates within genes and are particularly depleted within the NIST high sequencing confidence regions, as expected. However, given that the NIST high confidence regions cover most of the genome, there are still many millions of systematic biases across these high confidence regions in absolute terms, including within clinically important panels. Now that you know how my method works and understand its potential, you might be wondering how broadly this method could be applied by yourself or by colleagues who rely on accurate sequencing data. We have developed two main approaches for users to do this. You can read summaries of these on our poster, or get more detailed information from our publication in Genome Research or the associated preprint, the links to which are also included on the poster. There are also many possible ways you can adapt this method for similar situations. For example, I recently generated several incremental databases for coronavirus sequences generated at the University of Sheffield Sequencing Centre in order to check data quality and to establish optimal sequencing parameters. Using these, I was able to discover with a colleague that a specific variant called S943P in the coronavirus spike protein that was of interest in an earlier version of the bioRxiv preprint on the left was in fact a false positive, and luckily the false positive variant was removed from the preprint just before the story got picked up by the international press, saving us a lot of embarrassment and demonstrating once again the value of our method. This work has now been accepted for publication in the journal Cell. In summary, what are the main impacts from this work? Firstly, we have identified that systematic sequencing biases are widespread within Illumina HiTSeq whole genome sequencing data and lead to false positive variant calls, including within clinically important regions. Secondly, we have catalogued these systematic biases and provided tools for researchers and clinicians to use so that they can avoid false positive variant calls in their results arising from these. Finally, we have published this work and the associated tools in Genome Research, and you can also find a very similar preprint at this link here. If you have any questions, please address them to tmfreeman1@sheffield.ac.uk, and I will respond to you as soon as I can.